carbohydrates, if they contain more than one unit, monomeric unit, then these are held together by the glycosidic bonds. What is their importance in the body, or why these carbohydrates are important? It is important because it supplies the energy to the living organism, and also it acts as a structural constituents. Okay, so this is just briefly the definition, or how these carbohydrates are briefly described. Okay, now carbohydrates, these are the most abundant organic molecules in nature. So if you see the molecules which are present in nature, the most important organic molecule in nature, it is the carbohydrates. Okay, primarily they are composed of the carbon, hydrogen, and oxygen. So the main constituent of the carbohydrate, it is the carbon, hydrogen, and oxygen. Because of this, initially they are also called as the hydrates of the carbon. That is, hydrates, you know it is the OH group. Okay, so OH group attaches to the carbon. Okay, so mainly the main constituents of the carbohydrates, it is the carbon, hydrogen, and oxygen. And since this oxygen and hydrogen, it forms the hydroxyl group, it is attached to the carbon. Because of that, initially they are called as the hydrates of the carbon. Okay, so later on we also know that the carbohydrates, it also contains the other elements in addition to the carbon, hydrogen, and oxygen. And this may not be the hydrates of the carbon. The carbohydrates may not be the hydrates of the carbon. There may be some other carbohydrates which we will not call as the hydrates. Okay, so we'll see that one later on when we discuss more details about the carbohydrates. Now, some of the carbohydrates, it possesses the empirical formula, that is CH2O within bracket and 2N, okay, where the N is 3, satisfying that they are in fact the carbohydrates, that is the hydrates of the carbon. Okay, so this CH2O, this one N, then N stands for the 3. That means there should be 3 numbers of the carbon, there should be 6 number of hydrogen, and there should be 3 numbers of the oxygen. So this should be the minimum formula for the carbohydrate. They should satisfy this formula. Okay, then however, there are several non-carbohydrates which also appears as hydrates of the carbon. So initially I have mentioned that the carbohydrates, they are initially defined as the hydrates of the carbon, but later on it was observed other molecules which are not carbohydrate, but it is the hydrates of the carbon. Okay, and moreover, it fulfills this empirical formula. So that means now these carbohydrates we cannot define as the hydrates of the carbon. Okay, so however, there are several non-carbohydrates which also appears as the hydrates of the carbon, like the acetic acid, lactic acid. Okay, some genuine carbohydrates, that is the rhamnohexose, deoxyribose, they do not satisfy the general formula, the empirical formula given, that is CH2O and N. So these molecules, okay, the rhamnohexose and deoxyribose, it does not satisfy this empirical formula. But we know that these are the carbohydrates. That means this empirical formula given earlier, it is, now we cannot say that all the carbohydrate it should fulfill this empirical formula. Okay, so 2 things which we have find out here is that there are some compounds which are the hydrates of the carbon but not the carbohydrate. Because of that, we cannot call it as the carbohydrates as the hydrates of the carbon. Secondly, there are some genuine carbohydrates which does not fulfill this empirical formula given for the carbohydrates. Okay, so carbohydrates, it may be defined as the polyhydroxyaldehydes or the ketones. Okay, that means it is the polyhydroxy ketones or the compounds which produce them on hydrolysis. So how we define the carbohydrates? So carbohydrate is that they should be polyhydroxyaldehydes or polyhydroxy ketones, or any compound on hydrolysis. If the polyhydroxyaldehydes or the polyhydroxy ketones are produced, then we also call it as the carbohydrates. Okay, simply you can take the example of the polysaccharides like the glycogen, starch, this and all that. Okay, so all that polysaccharides, they will not be fulfilling this definition, that is they are the polyhydroxyaldehydes or the polyhydroxy ketones, because the functional group, the aldehyde or ketonic functional groups, they are involved in the formation of the bond. So they will not be free. These functional groups will not be free. So So these compounds, so if you see it individually, when it is in the polymeric form, then it is not going to fulfill the definition of the carbohydrates. But once these compounds are hydrolyzed, then they are going to produce the products which fulfills this definition, that is the polyhydroxyaldehydes or the polyhydroxyketones. So these compounds which on hydrolysis produces this molecule, they are also classified as the carbohydrates. Okay, examples are the polysaccharides. So you may take the cellulose, you may take the inulin, okay, glycogen, starch, etc. Then another term, the sugar, it is applied to the carbohydrates soluble in water and sweet tryptase. So there is a group of the compounds among these carbohydrates which are soluble in water. So many of the carbohydrates are not soluble. Say if you take the cellulose, okay, if you take the cellulose, this will not be soluble in water. So like that, there are many carbohydrates which are not soluble in water, but there are some carbohydrates which are soluble in water, and if the taste of these carbohydrates is sweet, then we use the term sugar for this type of the carbohydrates. Okay, so before we go to the carbohydrates, that we need to know that why these carbohydrates are important. Okay, so important means what type of functions these carbohydrates perform in our body. It may be in our body, it may be in the animals, it may be in case of the plants. So that means it is the function of the carbohydrates in the living organism. So what are the important functions? So important functions performed by one, they are the most abundant dietary source of energy, and it produces about 4 calories of energy per gram for all the organisms. So if these carbohydrates are digested or if they are oxidized in the living organism, then the amount of the energy produced is equal to 4 calories per gram in all the living organisms. Secondly, they are the precursors for the many organic compounds like the amino acids, fats, etc. Okay, it may be discussed in details when we discuss the metabolism. They also serve as the structural components of the many organisms, like the exoskeleton of the sun insects and a cell wall of the microorganisms. Okay, so in addition to this, we have also discussed it when we discuss about the biological membranes, that some of the carbohydrates, they are present on the outer surface of the plasma membrane, and they act as a receptors. Okay, they are act as the receptors. It may be for the cell recognition, it may be the receptors for the hormones, it may be receptors for the other things. Okay, so many carbohydrates, they are present on the cell surface or in the plasma membrane of the cells, and they perform a specific function in the cells. Okay, fourth, they serve as the storage form of the energy. Okay, so energy, say suppose we are taking the food now. Okay, so this what we are taking now, it has to be reserved. Say we are taking now after 2 hours, then we may need the energy. So from where this energy will come? Energy will come from the stored energy. Okay, so these carbohydrates, they also act as a storage form of the energy. The example is that glycogen in case of the animal, starch in case of the plants. Okay, to meet the immediate energy demands of the body. Okay, so these are broadly the functions that are performed by the carbohydrates. Okay, any question till now? Do you have any question in this area which we discussed it?
No, sir.
No. Okay, so we'll move ahead. Uh, I'll take your class up to only 12, because from 12 we have another meeting, so I will finish at 12. We have 26 minutes. Okay, then classification. So carbohydrates, they are broadly classified into 3 groups. That is the monosaccharides, oligosaccharides, polysaccharides, based on the number of the sugar units present. Okay, so the monomer, based on the monomer unit present, then all these carbohydrates we can classify into 3 groups that are the monosaccharides. Means these are the simplest form of the carbohydrates. They cannot be further degraded. Okay, they cannot be further degraded, or they may not be further hydrolyzed to produce the carbohydrates. So they are the simplest form. Then oligosaccharides, these are the carbohydrates which contains from 2 to 9. Okay, So if the number of the monosaccharide units present is 2 to 9, then we classify it as the oligosaccharides. There are some other classifications that some people, they also classified as the disaccharides and then oligosaccharides. But all that disaccharide, trisaccharide, this all comes in the oligosaccharides. So it's better if we classify it as the monosaccharides and oligosaccharides. Okay. Lastly, it is the polysaccharide. So poly means many. So if the number of the monomeric units is more than 9, so up to 9 we have defined it as the oligosaccharide. If it is more than that, it will come in the polysaccharides. And generally, the number of monomeric units present in polysaccharides, it is in thousands. Okay. It's a very large number of the monosaccharide units are present in the polysaccharides. Okay, monosaccharides and oligosaccharides, they are sweet to taste, okay, and they will be soluble. These monosaccharides and oligosaccharides, they will be soluble in water. They have the sweet taste. They are crystalline in character. Okay, hence they are commonly known as the sugars. So earlier we have defined water as sugars. So sugars means it is if they are dissolved, if they can be dissolved in water, and if they have the sweet taste, we classify it as sugar. So here, the monosaccharides and oligosaccharides, they are soluble in water. They have the sweet taste. So this we classify as a sugar. Okay, but if we take the polysaccharides like the starch, okay, so initially this will not be sweet in taste. Okay? Later on, when we digest it, then this the starch or the glycogen, they will be degraded to form the monosaccharides, oligosaccharides, and all that. So they may have the sweet taste, but initially, before its digestion, they will not have the sweet taste, so they will not be classified under the sugars. Okay, now let's come to the monosaccharides. So monosaccharides, it is the word derived from the Greek. Mono means one. Okay, so that means these are the carbohydrates which contains only one unit. Okay? So they are the simplest group of the carbohydrates and are often referred to as a simple sugars. Okay, so here they are the simplest group among all the carbohydrates, and they are referred as the simple sugars. Okay? They have the general formula, that is the CnH2ON, and they cannot be further hydrolyzed. If they are further hydrolyzed, they will not become the carbohydrate. Okay, so they cannot be degraded further to produce the carbohydrates. If they are degraded, they will no more be a carbohydrates. Then further, these monosaccharides, they are divided based on the functional group. Okay, so in the definition we have defined that they may contain two types of the functional group. One is the aldoses, and another one is the ketoses. Okay, so those carbohydrates or the monosaccharides which have the aldehyde functional group, it is called as the aldoses, and those which have the keto functional group, then they are called as the ketoses. So based on the functional group, they are classified as the aldoses or the ketoses. Okay, then the carbohydrates, it also contains some of the carbohydrates, it may have the 3 carbon atoms and maybe have 4, 5, 6, 7, and all that. Means the number of carbon atoms present in these monosaccharides, it varies. So based on the number of the carbon units present in these molecules, they are further divided into trioses, means containing the 3 carbon atoms. Okay, if the monosaccharide contains 3 carbon atoms, we call it as a trioses. If 4 carbon, they are called as the tetroses. If 5 carbon, pentoses. 6 hexoses, and 7 heptoses. Okay, heptoses, they are very rare, and it may be found only as a intermediate of the metabolism. So when the carbohydrates are metabolized, then some of the carbohydrates, it may be containing 7 number of the carbon atoms. Okay, we'll come to know it later on when we discuss the metabolism. Okay, so here the aldoses, when the functional group is monosaccharide, it is an aldehyde. They are known as the aldoses. Example we have given as the glyceraldehyde. Okay, then ketoses, the keto functional group. So the example is the dihydroxy acetone. We will come to these structures later on. Then oligosaccharides. So oligosaccharides, here it is oligo means few containing 2 to 10. Okay, I need to correct myself. I have mentioned as 2 to 9. Here it is mentioned as the 2 to 10. Okay. So these are the carbohydrates which contains 2 to 10 monosaccharide molecules. Okay, so since these molecules contain 2 to 10 monosaccharide units linked together by the glycosidic linkage, if they are hydrolyzed, then it will liberate all that monosaccharide units. Okay, so if that carbohydrate contains 2 monosaccharide units on hydrolysis, it will produce the 2 monosaccharides. They we call it as a disaccharide. Okay, if that contains 3, then we call it as a trisaccharide, and on hydrolysis, this will produce the 3 molecules of the monosaccharides. So here, based on the number of the monosaccharide units present, oligosaccharides are further subdivided as disaccharides, trisaccharides, tetrasaccharides, pentasaccharides, etc. Means depending on that the number of monosaccharide units present in these carbohydrates. Disaccharides produces 2 monosaccharides on hydrolysis. Examples are, is a very common one, maltose, lactose, sugar. Okay, so maltose, then lactose, sugar. These are the most common disaccharides that are present in nature. Trisaccharides example is the raffinose. Okay, which is formed by the combination of 1 molecule of fructose, 1 molecule of galactose, and 1 molecule of glucose. Tetrasaccharides, that example is the xylose, then pentasaccharides, it is the verbicose. Okay? So these are the examples, and this is how the oligosaccharides are further classified. Polysaccharides, so poly means many. Okay, so are the polymers of the monosaccharide units with a high molecular weight, so it may contain up to 1 million. Okay? They usually are tasteless and form the colloidal water. So that is what we said, that these polysaccharides initially they will not have any taste because they are not the sugar. Okay, then they are not purely soluble in water, but it forms a colloid with the water. Okay, there are 2 types. One is the homopolysaccharides, means it is also called as the homoglycans, and here if the monosaccharide unit is same. Say suppose all the monosaccharide or the monomeric unit it is glucose or fructose, then it will be homopolysaccharide. Means it is same or only one type of the monosaccharide is involved in the formation of the polysaccharide. Okay. Homopolysaccharides contains the monosaccharide units of a single type and may be represented by the general formula. That is, the examples are starch, glycogen, inulin, cellulose, etc. Okay, starch, glycogen, cellulose. Here the monomeric unit is the glucose. In case of the inulin, the monomeric unit is the fructose. Okay. Then second type, it is the heteropolysaccharides. So hetero means it is different. So that means it possesses 2 or more different types of monosaccharide units or their derivatives. Okay, so here more than one type of the monomeric unit is present. Examples are heparin, chondroitin sulfate, etc. Okay, next is the stereoisomerism in monosaccharides. So you must have already studied in your chemistry class what are the stereoisomers and all that. Okay, so stereoisomers are the compounds that have the same structural formula but differ in their special configuration. Means these are the molecules which have the same chemical formula. The formula, structural formula, it is same, but how the groups are arranged in space in that molecule is different. Okay? The arrangement of the groups in the molecule it is different. So that means the structural arrangement of the special arrangement of the groups in the molecule it is different. Okay? Then a carbon is said to be asymmetric. Okay, so we have the asymmetric carbon, symmetric carbon, and all that. Okay, asymmetric, that means when the carbon has 4 different groups, means the 4 different groups are attached to a carbon, then we call it as an asymmetric carbon. Okay, a carbon is said to be asymmetric when it is attached to 4 different atoms or the groups. The number of asymmetric carbon atoms determines the possible isomers of a given compound, which is equal to 2 to the power n. So in a molecule, that how many isoforms or the isomers will be present, it depends on the number of asymmetric carbon present. Okay, so this is equal to 2 to the power N. Say, in case of glucose, the number of asymmetric carbon atoms present is 4. So 2 to the power 4. Okay? That means it will have 6 isomers. Okay? So number of isomers, it will depend on the number of the asymmetric carbon atoms present in the molecule. Glyceraldehyde is the simplest monosaccharide with one asymmetric carbon atom. When I think we'll come to the structure of the glyceraldehyde. So this is the simplest carbohydrate, and it has one asymmetric carbon. Okay. Then it exists as 2 isomers because 2 to the power 1. So it will be 2. Okay. So it should have 2 types of the isomers. It exists as the 2 isomers and has been chosen as the reference carbohydrate to represent the structure of the all other carbohydrates. So when we assign the structure that DL to the carbohydrates, then the glyceraldehyde is taken as a reference compound. So glyceraldehyde, it occurs either in the D form or in the L form. Okay, those carbohydrates which resembles the structure of the D will be called as the D sugars, which resembles the L glyceraldehyde will be called as the L series. Okay, the D and L isomers are the mirror images of each other. The special orientation of the hydrogen and hydroxyl groups on the carbon atom that is adjacent to the terminal primary alcohol determines whether the sugar is D or the L isomer. Okay, we'll come to this here. Okay. Now you have the, is this one crusher visible? This crusher which I'm putting in the figure, is it visible?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So let's see this here. We have the 2 molecules, that is the D-glyceraldehyde and L-glyceraldehyde. Okay. So here, if you see the carbon number 2, okay, the middle carbon, then for this molecule, the OH group is on the right-hand side. Okay. So when this OH group is on the right-hand side, we call it as the D. The configuration for this, it is the D. Now, when the OH group attached to this asymmetric carbon, it is on the left-hand side, we call it as the L. So D-glyceraldehyde and L-glyceraldehyde, it is based on the arrangement of this group, that is the hydroxyl group and hydrogen atom attached to the asymmetric carbon. Okay, so this D-glyceraldehyde and L-glyceraldehyde is taken as the reference compound for assigning the configuration of the other carbohydrates. Okay, here let's take the example of the glucose here. Okay, so if you see here, then this glucose molecule, it has 4 asymmetric carbon, that is carbon number 2, 3, 4, and 5. Okay, carbon number 2, 3, 4, and 5, these are the asymmetric carbon atoms. So to assign the configuration of this glucose molecule, what you have to do is that you have to see the configuration of the OH group and hydrogen attached to the carbon atom, the last asymmetric carbon atom. Okay, that is the carbon number 5. Okay, that is the asymmetric carbon adjacent to the terminal carbon. Okay, so that is the carbon number 5. Now, if you see here, the OH group is on the right-hand side. So that means this glucose molecule will be calling as the D-glucose. Okay, if you see the other molecule here, then the OH group attached to the last asymmetric carbon, it is on the left-hand side. That means it is resembling to the D-glyceraldehyde. So this will be called as the L-glucose. Okay, so how the D and L configuration is assigned to the carbohydrates, it has to be done with reference to the glyceraldehyde. If it resembles to the glyceraldehyde, we call it as the D sugar. If it resembles to the L-glyceraldehyde, we call it as the L sugar. Okay. So for glucose, C5 is the carbon atom adjacent to the terminal primary alcohol carbon, and this determines whether the glucose is D or L isomer. Naturally occurring monosaccharides in the mammalian tissues are mostly of D configuration. Okay. So whatever that monosaccharides or the carbohydrates that is present in the living organism, especially the mammalian system, it is of the D form. Okay? This is just because that the enzyme system which are present in our body. Okay, the enzyme system present in the mammalian system, they are specific for the D sugar. So since they are specific D sugars, they will be acting on these D sugars, or when they synthesize the carbohydrates, they will be synthesizing the D configuration, the sugars. Okay, so because of that, most of these sugars which are present in the mammalian system is of the D configuration. But when we discuss about the amino acids, we'll find that the amino acids which are present in the living system, it is of the L configuration. So all this D configuration will occur, or the L configuration will occur. It all depends on the enzyme system present in the living organism. Okay. Next, optical activity. It is a characteristic feature of the compounds with the asymmetrical carbon atom. So if a compound contains the asymmetric carbon atom, then they are optically active. Okay, they have the optical activity. That means they can rotate the plane polarized light. So the solution is kept. If a plane polarized light is passed through this solution, then this plane polarized light, it is either rotated on the right hand side, that is in the clockwise direction, or they are rotated in the anti-clockwise direction. Okay, so this the terms dextrorotatory, or it is also called as the plus sign, or the small letter D, or the laboratory, represented by minus sign or the small l. These are used to compounds that respectively rotate the plane of polarized light to the right or to the left side. So if it is rotated in the right side, okay, that means in the clockwise direction, it is called as the dextrorotatory. And if that is rotated in the anti-clockwise direction or in the left-hand side, then we call it as the laboratory. Okay, now this is the family of the aldoses. So I think we'll stop here. I need to go for the meeting, so we'll stop here. Next time we'll discuss with this one, that the family of the aldoses, or the structure of the monosaccharide. So here, if you find it here on the top, it is the smallest one, and then it goes down to the molecules which contains the 6 carbon atoms. Okay, 1, 2, 3, 4, 5, 6. So these are the hexoses, and I have mentioned that the heptoses, which contains this 7 carbons, they are very rare in nature, and they are produced only as an intermediate of the metabolism. But these are the ones which are commonly found in the nature. Okay, so we'll discuss these structures maybe in the next class. So if we start with the glyceraldehyde, okay, that is the triols, then you have the tetrols, pentoses, and hexoses. Okay. Same thing for the ketoses. Now you have the triols here, that is the dihydroxyacetone. Okay, if we see here that the dihydroxyacetone, it doesn't contain the asymmetric carbon, so they will not be optically active. Okay, if you go to the erythrulose, then it contains one asymmetric carbon, that is carbon number 3, so they will be optically active. Okay, so this is the triols, tetrols, pentoses, and the hexoses. Okay, next. We'll come to this epimer and all that. Okay, maybe we'll start with aldoses, then that ketoses also in the next class. So let's stop here for today.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.